Hello, everyone. My name is Ian McLaughlin, and I have a PhD in neuroscience. And today I wanted to talk about a fun study that explores a question that I think a lot of us wonder at one point. What is the best way to remember something that we're trying to learn? How do we optimize the process of learning? And so this is a group out of the Max Planck Institute of Neurobiology in Germany, as well as the Ludwig Maximilian University in Munich. And what they're interested in is an effect called the spacing effect, which is basically the dynamic that I think is somewhat intuitive to many of us, where if you add a few breaks, during an extended length of time when you're trying to learn something new, it seems like we tend to experience both strengthened memory as well as greater longevity of the memory, right? In other words, we learn things more effectively when we take breaks. So if you're short on time, that's the takeaway. This group found a whole bunch of evidence to support that taking periodic breaks when learning a specific thing, along with repetition, results in better memory retention. But if you want to understand why, maybe you'll stick around. So probably the most classic trade-off that many, if not most of us, have had to confront in the past, probably, when we were studying for tests in school or in college or whatever. And, we know we haven't 100% nailed all the stuff we need to know for a test or something. Do we stay up all night and cram for the test? Or do we just study as much as we can and get a good night's sleep, right? Now that whole experience is kind of related, though that whole situation implicates a bunch of other processes like the role that sleep plays in consolidating memory and sustaining cognitive acuity and stuff like that. But this whole process is happening while we're awake. And so there's something that happens while we're awake, when we take a break, while we're trying to learn something, that seems to amplify our ability to retain what we learn over the long term. However, there is a trade-off. So let's get a little bit more specific here. So let's begin at the start. When we learn something, a specific constellation of our approximately 86 billion neurons is activated, right? Just like when somebody plays a piano. If you hit one constellation of those keys, you get one chord. If you change just one of those keys, you get a different chord, a noticeably different chord. Now, even the fanciest and most beautiful of grand pianos have about 88 keys, right? There's some pianos with more than 88 keys, to be clear, but they're kind of rare. And of course, there's always iterations on a similar theme with far more keys. So this is, by the way, all of these sort of yellowish pictures. This is the largest organ in the world. It's absolutely beautiful. And do you know how I know it's beautiful? It just happens to be a few blocks from where this camera is pointing right now in Philadelphia. So this beautiful, majestic thing is languishing in like the frivolity of, a mall. But it's called the Wanamaker organ. And this bad boy has 6 manuals, 28,750 pipes, 464 ranks, and a whole ton of stops and controls that modify the sounds it makes. It also has 88 ranks of string pipes. It basically fills a beautiful 7-story building right next to City Hall. And it's the kind of thing that's so large and so complicated and so powerful that you can't help but kind of have an instinct that it's maybe sentient. Anyways, it's a really cool part of the city that I think a lot of people, even who live here, don't know is here. So I don't know anything about this kind of instrument, right? An organ. I've never played one. But compared to a piano, I know that, that's quite a few more keys than the typical pianos I've encountered. Keys, pedals, stops, pull knobs, all kinds of ways to produce sound. And I know different organs have different numbers of keys, but I can tell that this beautiful instrument has an intimidating number of keys. Look what this guy's sitting in front of. More keys than I'd know what to do with, for sure, including the pedals that are at the feet. It's crazy. That said, if the standard piano typically has 88 keys, compared to the number of neurons in the human brain, the potential combinations of keys that could be played at the same time, which is quite a lot on a piano, is essentially negligible compared to the potential combination of neurons that can and are activated in the brain, right? 88 keys on a piano is a lot, don't get me wrong. It's typically the largest keyboard out there that people play, right? But we have somewhere around 86 billion neurons, and around as many of another type of brain cell called glia, all of which are doing something every millisecond that we're alive, right? And so, okay, did you just zone out? Well, even if you're zoning out and not focusing on anything, right? Millions and millions of cells in your brain are being activated every millisecond. And even with those beautiful instruments, with their tremendous number of keys, right, it pales in comparison to the number of possible combination of neurons that can be activated at the same time in the human brain. And so, when we're learning something, when we're exposed to something new, specific constellations of neurons are activated. So imagine each of these little blue circles is a neuron, okay? Well, including the red one as well. So imagine each of these circles is a neuron, and the secret sauce of our neurons is that they can influence each other's activity, right? And so at a minimal level, imagine that if you activate one of those circles, let's say the red one, the other circles are activated or influenced in some way. And so that's sort of what we're talking about. And of course, each of those networks of neurons is embedded in an almost incomprehensibly complicated network of other networks, right? And so, so similar concept here, right? But this is a far more complicated network of neurons relative to this one, right? But, so you might start to appreciate that many of those circles are likely involved in multiple networks of neurons, not just one. And so when you activate one constellation, another constellation might get a little tickle of activity because some of the same neurons are involved in both, right? So anyways, this is kind of like a dramatic, almost offensive oversimplification. But the point is that, when we learn something new, a specific constellation of neurons is activated. And the memory of that experience is thought to result from either the formation or the new formation of new connections between neurons, right? New lines between these various circles, or just the strengthening of, existing connections, of existing constellations. And then moving forward, right? That's, when you first learn something new. Moving forward, that memory becomes consolidated, or made stable over the long term, so it lasts longer. And this occurs by more extensive physical changes that occur in the brain. New synapses form, old synapses are fortified, and that whole process takes time. But the end product is a long-lasting memory. Okay, but for this question, right, how do we improve our ability to learn new things? We're not focusing specifically on the process by which we learn new things, which is a really interesting topic. Today we're focusing on a particular thing that seems to improve our ability to learn new things. The spacing effect, right? And so this group up here, Max Planck, the group in Germany, had a certain hypothesis. So here's a quote from one of the scientists. The molecular and synaptic mechanisms underlying the spacing effect can influence two characteristics of neuronal ensembles. Think of these as ensembles, i.e., the size or reactivation pattern of the ensemble during memory encoding, storage, and retrieval. So in other words, they assumed that by taking breaks during the process of learning something new, either the size of these constellations of circles, neurons, or the composition of the constellation itself is subject to change, right? So the rationales underlying those two possibilities are a little complicated, right? But they basically revolve around the expectations that, following multiple exposures to something we're trying to learn, those microscopic changes that occur in our neurons, during the first exposure to the information is sort of extended in the following exposures. You're sort of repeating that same activation pattern. And so, that might sound less intuitive than it likely is for most people, right? You might think about it sort of like this, right? I know this is sort of a strange comparison. I don't know why it came to me, but just stick with me. So, you're exposed to new information and you begin the process of learning that new information. But that initial exposure really isn't enough to sustain the information, right, in your memory. It's likely to just fade away for a variety of reasons. Just if I were to toss a basketball onto my finger and, spin it one time, it'll topple over, pretty quickly. But if you have that initial exposure, you take a break, but then you come back to it, engaging the same constellation of neurons after some time has passed, you can sustain that neuronal activity. The same pattern of activity that occurred, not exactly the same, but pretty much the same pattern of activity that occurred initially. Just like this talented person periodically spins the ball on their finger, right? Keeping the spin going and preventing the ball from toppling over, like it would immediately if I were to try. Okay, so not an ideal metaphor, I know, but I'm going to subject you to the same metaphor for the other potential explanation, right, that this group had. The repeated exposures to learned information with breaks in between strengthens the neural connectivity underlying the learning that's taking place, right? Just like the repeated spins of the ball, right, with breaks in between, right? He's not just like constantly touching the ball, right? He's taking little breaks in between, keeps that ball spinning. Okay, so I feel like the basketball metaphor might actually make more sense with the second explanation that they have for why it is that taking breaks when learning a new thing amplifies our ability to learn that new thing. So there's more to why it might be that taking breaks while learning a new thing might be most compatible with how our brain works. And so here's a quote from their publication. Quote, "Sparseness would safeguard the specificity of the represented memory, whereas stronger connectivity would render the memory more resilient to homeostatic mechanisms that can result in forgetting and thereby increase the probability of retrieval." Okay, I know. So what they mean by that is less frequent activation of the neurons involved in learning something new might cause them to be less vulnerable to being sort of diluted in the connectivity that they ultimately form, okay, which enable them to focus more on the task at hand, learning this new thing. And then just the sequential exposures to a given thing, one is trying to learn, simply strengthens the connectivity between the neurons that have been activated during each exposure. And so, that's the fundamental question and concept behind what this group is exploring. Why it might be, if it's true, that taking periodic breaks while learning something new seems to amplify our ability to learn that new thing. So here's what this group did. So first, they were working with our very close mammalian ally, the mouse. I know it might sound a little strange to call mice, almost universally experienced as a pest, an ally. But the reality is that essentially all meaningful biomedical research relies so heavily on the mouse that without them, that research would be very severely crippled. I worked with them. The most brilliant scientists I've met work with them, and we should all be grateful that mice are around. But so okay. Anyways, what this group did was explore a matching to place task. And so ultimately, the mice had to remember where a buried piece of chocolate was in a certain kind of maze. And so this is called the radial arm maze. And so as you can see, this is a maze with a central area that presents essentially 8 equivalent options for the mouse to explore, right? There's nothing meaningfully different about these arms, except, when it comes to this experiment. One of the arms has a bit of chocolate that they've been trained to remember to find, right? And so their whole setup was a little bit more complicated than this picture. This wasn't the maze that they were using, but it'd likely look quite a bit like this. And, I suspect there'd be walls on every side of the arm, of every arm, and obviously a little dish of something through which the mice could dig to find the chocolate, right? The chocolate was covered up. And so, mice were trained to know where the chocolate was. And so funny enough, I actually did a very similar experiment, a very similar experiment actually, but I went with Reese's Pieces rather than chocolate. Literally zero good reason beyond that I spoke to another grad student at the time, and her mice responded really well to Reese's Pieces, so I went with that. My mice loved them too. I have some pretty cute videos of them like munching on a little Reese's Pieces or a little Reese's piece. But my experiment didn't bear any fruit. But so anyways, the scientists were they trained these mice to remember where the hidden chocolate was in these radial arms, right? And they were trained 3 times, okay? They had 3 opportunities to explore the maze to discover this, rewarding thing that was, available to them. And so 3 opportunities to discover where the deliciousness was. And so here's a quote from the lead scientist. Quote, "Mice that were trained with the longer intervals between learning phases were not able to remember the position of the chocolate as quickly, but on the next day, the longer the pauses, the better was the mice's memory." So that's the sort of crux of this study. Longer pauses between training sessions equated to more effective discovery of the chocolate later on, okay? That's a fundamental takeaway. And so the group did a lot more than just assess how the mice performed. They used a whole variety of techniques to get a sense of what was actually going on in the brain while all this was going down. By the way, using many of the same exact techniques that I used in my research, like down to the same viruses or types of viruses. Don't worry, I'm not going to get to that level of detail because it's unnecessary to extracting some nutrition, some deliciousness from the work that this group did. But one of the cool things that they did was to measure changes in the activities of neurons in the prefrontal cortex, which, as you can see here, is basically the part of the brain that's as close to your finger as possible if you just put it like right between your eyebrows, like right there. As the brain gets to your forehead, right? That's about as close as the brain gets to your finger, right? Or certainly the prefrontal cortex. So it's the PFC, the prefrontal cortex, is sort of a broad anatomical region, and it's involved in a ton of aspects of consciousness and behavior. It's uncontroversial to say that it's one of the main brain regions that's like central to what it is in our brain anatomically that distinguishes humans from all other animals, as well as other primates, right? Chimpanzees, bonobos, and so on. It's clearly a pretty large region, right? You can see this red area is quite large. It's involved in everything from our ability to plan future actions, making decisions, controlling our more primitive impulses, like not eating an entire pint of ice cream, even though it tastes really good. These types of things. Robert Sapolsky describes it as the part of the brain that enables us to do the harder thing because we know it's right. As always, the team did quite a bit more than I'm going to discuss today. They did all kinds of variations in that memory test, with the maze. Things like not keeping the chocolate on the same arm for every trial, and placing the mice in different points in the maze as opposed to like right in the center every time, to get a sense of how they were developing their memory of where the chocolate was. Like, were they relying on a memory of the maze itself, a world-centric or allocentric reference, or were they relying on a memory of where they were in the maze, a body-centered or egocentric reference, right? So like, on what were they basing their memory? A bunch of really interesting questions. But the central question was, again, does inserting a bit of break time between learning a thing influence how effectively we learn that new thing? The answer is yes. Inserting some time between sessions of trying to learn a thing resulted in these mice more effectively learning the task. But not only that, the reactions of neurons in that part of the brain, the prefrontal cortex, and actually, it's actually a specific region of the prefrontal cortex called the dorsomedial prefrontal cortex. So towards the top of the PFC, towards the top of the prefrontal cortex, and then also towards the middle, towards the midline, dorsomedial. And so that's where they were focused, and they were focused on that part of the brain that was activated during this task. And what they found was that these neurons were sort of more unified when there was more time between each of those learning experiences. Okay, so in other words, when the mice had a bit more time between sessions, learning sessions, when they were learning where their chocolatey treat was in that radial arm maze, the neurons in the part of the brain that some neuroscientists call the prefrontal cortex, that in mice, that seem to be centrally involved in that learning process, were even more engaged. More break time, better engagement by these neurons, or more coherent engagement by these neurons. And so here's another quote from one of the scientists. Quote, if three learning phases follow each other very quickly, we intuitively expected the same neurons to be activated. Makes sense. After all, it is the same experiment with the same information. However, after a long break, it would be conceivable that the brain interprets the following learning phase as a new event and process it with different neurons. End quote. So basically, the team was expecting the opposite of what they found. They were expecting that if we encounter the same thing in rapid succession, essentially the same exact neurons would be activated each time. Totally makes sense. If I were to, tap your nose 3 times super rapidly, essentially the same neurons would be activated each time. And when it comes to something like a behavioral task, the less time that passes between each task, the less chance there might be for other factors to start to influence which neurons are activated, right? Things like hunger, mood, tiredness, energy, right? Even things like odors in the room, which is a massive factor to our little mammalian friends here, basically equivalent to vision for humans. All of those things would inevitably vary less when less time passes between trials, right? And all of those things, of course, would influence any animal's approach to learning something new, right? Despite the intuition that spacing out learning sessions tend to result in more effective learning. Honestly, I think I'd have the same hypothesis that this team had, that, at a certain point, too much time between learning sessions results in a disruption of effective learning, because it's just more likely that different neurons are going to become recruited at different times of day, let's say. Not that they had that much time pass, but yeah, that totally makes sense to me. But what this group found was that the longer breaks resulted in more effective learning and more coherent neuronal activity, which of course is basically saying the same thing in two ways, right? More coherent neuronal activity in this brain region is why the mice learn this task more effectively, or at least a large part of why, at least according to the data from this study. But the point is, the team not only replicated what has been known for a while, right? Taking breaks when learning something new seems to amplify our ability to learn that new thing, but they also identified what appeared to be at least part of the neural basis for that dynamic, for the spacing effect. Pretty cool. So if that's what you're most interested in, understandably, that's the takeaway, okay? Take some breaks when you're trying to learn something new. Not giant breaks, but some breaks. And by the way, that's absolutely 100% been my experience, particularly in graduate school, where I had to learn so much, so quickly for a test, right? That was completely new to me, particularly like gene regulation and stuff like that. I can't tell you how many times like I initially encountered something that I had to learn that initially struck me as being completely unintelligible and overwhelming and beyond my capability to understand. I had zero grasp of it, had no confidence that I'd understand it later. But after a bit of time, encountering that same information again, things just sort of fell into place a bit more readily, and I could move forward, right? So while we, should always, beware of findings that seem to ratify our intuitions in science, right? There are some interesting findings in this work, and intuition can sometimes be insightful when it comes to human behavior and consciousness. Okay, so takeaways. Take some breaks when you're trying to learn something new. That's the takeaway. And it seems that those breaks correlate with more coherent patterns of activation within the prefrontal cortex, specifically the dorsal medial prefrontal cortex. And so just sitting there, feeling guilty that you're incapable of spending, 2, 3, 4, 5 hours straight just studying something, you shouldn't feel that guilt. In fact, it's probably for the best that you don't do that. All right, so if you dig this kind of discussion, maybe you'll consider subscribing and liking and all that kind of stuff. That said, if you'd like to dig into some critiques of this study, you're welcome to stick around. So interestingly, this group used exclusively female mice. And that might sound kind of like a weird thing to even notice. And you're right, it is kind of weird. But there's been a pretty much a decades-long now concern within biomedical science and just biology that the vast majority of experiments that have that have been done have tended to use exclusively male mice for a variety of reasons that aren't really worth getting into now, but can be summed up by estrous cycle, basically the mouse version of the menstrual cycle. So, their explanation, this group's explanation, was that they did this to minimize intragroup aggression, to enable communal housing, which, fair enough. Totally the case, right? A bunch of, males that aren't related to each other, they will fight, and it can get really, really rough. Females will do it too, don't get me wrong, but males far more. And so fair enough, they wanted to do communal housing. But for me, I suspect there might be something else to this story. It has no effect on how I interpret these data, just to be clear, right, that they use exclusively female mice. It's just one of those things that you notice if you've done these kinds of experiments before. And, for the same reason that I think it's absolutely justified that more studies ought to be including both males and females, unless there's a good reason, unless you're specifically studying something in, female or male biology. This study doesn't seem to have a particularly good reason to not include equal numbers of both, right? It's just good practice. If you're trying to work with a representative sample, right, of participants in an experiment, you want your sample of participants to be as representative of the broader population that you're trying to understand as you possibly can. Go to extreme, lengths to try and achieve that. And so, okay, whatever. It's an interesting little quirk about this study. Another pretty controversial debate in neuroscience right now that's been going on for years. I've had this debate before, is whether mice actually have anything remotely approximating a prefrontal cortex to begin with, right? And so this is an entirely different topic. It's a fun topic. A lot of hot takes in the neuroscience world, particularly on Twitter. But I can assure you that this is about as spicy a debate in neuroscience as it gets, with some neuroscientists arguing that rodents simply don't have a prefrontal cortex, right? That there just aren't enough neurons in that neighborhood to meaningfully translate to the human or even the primate prefrontal cortex, right? There's some really good justifications for that perspective, to be clear, and some really substantive responses to that argument. And I linked to a paper in eNeuro, the journal eNeuro, in the discussion section below if anybody's interested in diving into that mire. It's a very dorky thing to get into if you're not conducting research, but it is just kind of like a fun example of how scientists vehemently disagree. Right? I think oftentimes we talk about science as though it's this unified institution, and it's just not, right? Scientists disagree all the time, in good faith, and this is one example of that. Oh, and by the way, the cool thing about eNeuro is that the articles tend to are open source, right? So you don't have to pay like several dozen dollars for a single article, which is just highway robbery, which is the case for the vast majority of peer-reviewed scientific publications. And what's extra cool about eNeuro, more interestingly, is you can actually read through the whole peer review process, right? It's pretty dense, don't get me wrong, but it covers the topic pretty effectively. And I think it might be interesting for somebody outside of the discipline to see how peer review goes. For anybody that's unfamiliar, peer review is the process by which your peers, your other scientists in your field or in related fields, scrutinizes a set of experiments that you want to publish. They try and find flaws, they try and find alternate explanations, interpretations, ways to improve the work, is sort of one of the best components of peer review. When it goes right, it's so fantastic, and I think that this is a fun one, the debate as to whether or not rodents have prefrontal cortex at all. And to be clear, the reason that's important to this study is that that's the brain region that this group honed in on. Is the prefrontal cortex. And so if mice don't have a prefrontal cortex, it doesn't mean that they're wrong about what they observed in those neurons. It's just, it might mean that they're wrong about translating it to our prefrontal cortex. Anyways, who cares? Okay, hey everybody. I'm sorry I didn't see your chats. Hey Scott, thanks for jumping in. Cosmic Rose, kiss and repeat, right? Heck no, drop the knowledge nuggets on us. I thought of you the other day when I heard someone say armamentarium. It's one of my favorite words, armamentarium. For anybody who doesn't, well, yeah, I'll just say, for anybody who doesn't know what an armamentarium is, that is sort of like, I guess, what is it? Like the artillery of a physician, right? It's like all the therapeutic tools at the disposal of a physician. And it's just this like overly, like, I don't know, belligerent term to be used for medicine, in my opinion. But I think it's really fun. And I always try and use it when I can. I find that I pick up, it looks like a complicated white Christmas tree stand to me. Yeah. This thing. Yeah, you're right. Probably could use it as a Christmas tree stand. So I find that I pick up more each time I go to the same material. That's normal, right? Yeah, exactly. Totally. Yeah. That's totally same for me too. And it's wild. Like even when I'm going back to a topic that I, ostensibly learned, I passed the test, I even got a good grade. But I haven't taken a test in many, many years. This is almost a decade ago now. So my memory may be failing me. But yeah, like, in synaptic transmission, things where there's like a bunch of different components that are necessary for a vesicle of neurotransmitter to be released, right? All these little proteins, each of which have their own little inscrutable names, have to interact with each other, twist together. It's this really remarkable process, by which, and it's happening in you right now as we speak, right? Dopamine being released, serotonin, norepinephrine, etc. They all essentially undergo the same process, which is, these little water balloons filled with neurotransmitters that have to fuse with the presynaptic membrane. Fuse with it, right? These little water balloons. So imagine if you took two water balloons and you put them next to each other and they just became one water balloon. And so that's how, you have neurotransmitter in one of the water balloons and then it becomes one water balloon and now the neurotransmitter is released, right? It's this really complicated process. Tons of proteins are involved. Anyways, I remember, passing that test, doing okay, and then coming back to it like several months later, maybe even a year later, and I had a completely different level of granularity of my understanding of it, appreciation of it. And I think that, that's a much longer break than we're talking about in this paper where, these were pretty short breaks, right? They weren't like many days between learning sessions. But still, I think, yeah, your intuition is exactly the intuition to which I was referring earlier. And so, M. Boyle, love these streams, keep them coming. Thank you for joining. Have you heard anything about Valve's collaboration with OpenBCI? Valve, like the video game company? I have not. That's pretty exciting. It'd be cool. It'd be really cool is if they, I did this. I collaborated very, very briefly with this company out of West Virginia that has made a virtual reality platform where you can explore microscopy data. In other words, like, imagine you get an MRI of your brain. And actually, I have a video of this pinned on my Twitter. It's been there for like many years just because I think it's so cool. But, you take an MRI, you have a picture of your own brain, right? You see where all the wrinkles are, where, your thalamus is, where your cerebellum is, where your prefrontal cortex is. And then, imagine that you are wearing a VR, right? Like a, Oculus Rift or something. And you're standing in a virtual chamber, your laboratory, whatever, or it's more like a warehouse. And then you have this 3D version of your brain, and you can manipulate it in virtual reality. You can make it bigger. You can make certain parts of it transparent, and so you can like literally walk along your ventral tegmental area, where a lot of your reward-related dopamine is produced. Not really reward, whatever, but they're trying to integrate a few different sensor types into their next VR headset. Okay, so they're just improving their own VR. That's awesome. I remember learning, early on when, there's no question that virtual reality is like so much better than it used to be, right? I remember hearing that sensory neuroscientists were being hired by a lot of these VR companies. Like I think Magic Leap, which I guess we haven't really seen much out of them in terms of consumer products. Or did they release? I don't know. I know, Oculus Rift, they hire sensory neuroscientists to sort of help them calibrate, it's a really difficult thing to do because you're essentially tricking the entire brain into believing that you're somewhere that you're not. And so, people get nauseous. It still happens in some cases, but you can mitigate that predisposition to nausea, by making the device as compatible as possible with your VR. EEG, EOG, EMG, EDA, heart rate, temperature, and wow, that's wild. Super cool. EEG, wow. This is going to be a complicated headset, but that's super cool. All right, so another thing about the study. This is even more granular and nitpicky, but if you really want to get a sense of what this group might be asked, right, if they were to, go to a different institution outside of Germany, let's say, where I went to graduate school, and present their research, right, which happens all the time, to an audience of scientists. One example of something that would be brought up is the type of mice with which they worked. Right, so without getting into the details, it turns out that we have different strains of mice, right? I mean, and you can observe this with your own eyes, right? You have white mice, you have black mice, you have brown mice, right? Those are different strains. In science, we use what are called inbred strains, so that let's just call them black mice. The name of the most widely used mouse in behavioral science, neuroscience, is C57 black 6J, and it's a ridiculous name, I know, but basically these are mice that are almost clones of each other. They're almost genetically identical. They're not, to be clear, they're definitely not. But they're far more closely related than what you might call, field mice or just wild mice that you might, see scampering around, right? There's going to be genetic diversity there, just like there's genetic diversity between humans. But so you have these strains of mice, and they're treated. It's a way for you to be able to control for any effective genes, right? So there's not some kind of genetic variation between the mice, and so therefore, if you see some kind of difference according to your treatment, it's unlikely to be due to some genetic difference between your mice. So you don't have to sequence the mice every single time, right? And so this group was working with a strain of inbred mice, and it turns out that these mice seem to have a retinal abnormality that potentially affects their eyes and therefore their vision, right? I'm almost certain that someone in the crowd of scientists listening to them present these data would ask about whether the vision of the mice might have had some effect on the performance in these memory-related tasks, where they had to discover the location of a buried, delicious treat, right? To be clear, I wouldn't be that scientist who would ask that question, but it is an interesting feature of the study, nonetheless, and it is a meaningful issue because, mice. How are mice finding this chocolate? They're using their sensory systems. Now, as I said earlier, vision is certainly not the most important sensory system to mice. Olfaction or their sense of smell is. And so I've seen no evidence that this strain of mice that they use have any kind of disruption of their olfactory system. But still, there are more nitpicky things that we could explore. And honestly, I know like 0 people would find that they would find that neither edifying nor interesting, right? And so this is just one study, and we can certainly look forward to seeing more groups interrogate this whole concept of the spacing effect at the level of the prefrontal cortex, and then the broader circuitry, because obviously, the prefrontal cortex is not the only brain region that's going to be involved in learning anything, right? It'll be important, but not the only one. But despite all these critiques, I think it's safe to come to a fundamentally similar conclusion that this group comes to, which is taking breaks while learning new things seems to amplify our ability to learn those new things. And so Cosmic Rose, I remember when you posted your VR clip on Twitter for us. I hope to be back on Twitter sooner than later. Cool. Wouldn't generations of inbreeding result in defects? Yes, Scott. Yes, you're exactly right about that. It's a sort of a fine balance, right? And so what Scott is emphasizing for anybody that isn't sure. Well, I think we all appreciate that inbreeding is not good. But why is it not good? Well, it's because generally speaking, right, when you have genetically very different people, right, humans only get that genetically different, by the way. We are by far the most inbred primate alive today. Isn't that crazy? Orangutans, bonobos, chimpanzees are far more genetically diverse than we are. Isn't that wild? Even though to my eyes, they all look very, very similar, right? I probably couldn't tell two chimpanzees apart. But for humans, and there's a lot of different, very mostly cosmetic, but very different things, different colors of skin, different hair colors, different textures of hair, right? Different amounts of hair, so many different eye color, different bone structures in the face, all that kind of stuff. But now we're very, very inbred as far as primates go. It's kind of funny. But anyways, so the problem with inbreeding is that if two organisms that are genetically almost identical mate, then it's possible for what are called recessive genes to become expressed, and sometimes recessive genes can be associated with insufficient production of a protein, or just disrupted, inefficient production of a protein. And so when you have genetic diversity, and let's say I have a recessive gene for whatever, I don't know, I have no idea, whatever, some kind of protein, right? The production of some liver enzyme. Let's say that if I have one copy of that gene that's recessive, right? But then I also have a dominant gene that produces enough of this enzyme, and then so as a result, I don't even know that I have that recessive gene because it just never becomes a problem. And let's say my wife also has a copy of that recessive regarding what they eat, because they might not be able to digest X, Y, or Z thing, right? So when it comes to these mice, you're exactly right. A lot of those genes are essentially identical. There is some variation, to be clear. They're not clones, and as a result, Scott, you do see some wacky stuff happen, right? One of the freakiest for me, and it is, it's not cool that it happens, but you get this sort of tusk-like effect, where for whatever reason the bottom incisors of the mouse, which are super sharp, right, they're that's what they use to cut into things, essentially, they just don't stop growing, and they look like tusks but pointing upwards. The problem is that if you look at a mouse's head, their nose extends well beyond their snout, extends well beyond their bottom jaw thing, right? And so if your teeth are pointing directly up and just continue growing, it can have all kinds of effects on their ability to eat, their growth of their skull. They become bradycephalic, I believe is the term. That's just one example. You can get really, you can get high anxiety, which is something that I studied. You can get a whole bunch of things, right? And so yeah, it's a peril. To be clear, that is very rare. That doesn't happen. That's by far, it's super unlikely. I'm sure many graduate students go through working with C57 Black 6J mice and never even see that. And it is easy to treat, just so to be clear. It is easy to treat. You just clip the teeth. But yeah, you're right. Yeah. And similarly, you can have different strains of mice that behave very differently. The reason that most behavioral neuroscience labs that work with mice work with C57 Black 6J mice is that they're very behaviorally active. They do a lot. They're very mobile. They're very action oriented. They can be pretty aggressive. So you get a whole behavioral repertoire. Whereas a lot of other mice are just sort of dumb. they're just sort of they're very nice pets, right? They're not aggressive. They're not super anxious. they'll just hang out, whatever. My experience, agouti mice are just, if I were to have a pet mouse, I won't, but if I were, I would go with an agouti mouse. Now, FVB mice, which are white mice, what I'm sure a lot of people think of when they think of lab mice, they are little terrorists. Like, you cannot work with them. They're so aggressive. Anyways, okay, that was a whole tangent I didn't expect to go on. But, okay, last thing, I'm not going to lie. One of the reasons I gravitated towards this study is that they used so many of the same techniques that I use in my own research, most notably the viruses and designer receptors that they used to control the activities of neurons in the prefrontal cortex. So we both used a technique called DREADS. Designer receptors exclusively activated by designer drugs. So basically, it's a neurotransmitter receptor. It can be any kind of neurotransmitter receptor. Opioid, mu-opioid receptor, actually kappa-opioid receptor, serotonin receptor, dopamine receptor, and so on, right? You can take any of those. This is a muscarinic receptor in this case. You can take one of those receptors and then you mess with it here and there, right? You change one part of the receptor so that it no longer is capable of binding the neurotransmitter that it normally binds. It can't bind serotonin anymore, right? It's just sitting there. Nothing's binding it. But is capable of binding a synthetic drug that you made that only binds that receptor and nothing else, right? And so to give you a sense of just how similar our projects were in this case, so this image, which is an image of the prefrontal cortex, right? That's the prefrontal cortex. Well, what they call the prefrontal cortex in a mouse. This image is other prefrontal cortex of neurons that they infected with their virus, the virus that drove the expression of their dread, right, their designer receptor. And so why is it useful to use these dreads? It means that you basically have your own pet receptor available in the brain to either activate, inactivate, or just influence the activities of specific neurons that you choose. Wherever you want in the brain. In this case, it's the prefrontal cortex. So, like, what happens if this specific, cluster of neurons or group population of neurons in this specific brain region, the prefrontal cortex in this case, what happens if you activate those neurons in a waking animal? How does it change their behavior? How does it change their ability to learn? And so, what happens if you turn those neurons off, which is what they did in this study? So it's basically a way for us to get an idea of what the various neural circuits in the brain actually do, right? By making certain neurons remote control. You can activate them, inactivate them. And so it's this really neat technology that I think deserves, a bit more attention than it gets. Okay. And so any new IPAs lately? Cheers to you. Yes. Well, not new, but this, I randomly stopped by this, this beer store, and they had Heady Topper and Vocal Banger. They had a bunch of it. And so now I'm restocked. I give out Heady Topper as gifts to people that I know are beer nerds, and Focal Banger. And so I had gotten down to like only 3, which I was going to keep for myself. But now I'm fully stocked again. So cheers. Pretty cool, huh? Maybe I'll have one tonight. Like a narwhal. Yeah, kind of. Well, it's really more like a walrus. You're talking about like the tusk situation in mice. They look kind of like a walrus, but an inverse walrus. But they're not. It's not as functional as it is for walruses. Walruses? Walrus? Is walrus like bear? Anyways, okay. Speaking of anxiety, did you ever study the DNA SNP of COMT? So did I ever study a SNP of catechol O-methyltransferase? For anybody who's unfamiliar, that's an enzyme actually in the prefrontal cortex, active in the prefrontal cortex. In the PFC, what it does is it does more than just this, but the main event is that it degrades dopamine. So dopamine is a catecholamine, which means that if you look at the molecule, it has a catechol. It's like a molecular shape, essentially. Norepinephrine, epinephrine, and dopamine are catecholamines, and catechol O-methyltransferase, by O-methyltransferasing, it degrades dopamine. And in fact, one of the interesting findings, well, there's 2. So one is in folks with schizophrenia, it turns out that certain SNPs are associated with certain SNPs, single nucleotide polymorphisms, genetic unique versions of that enzyme, of the gene that encodes that enzyme, are associated with schizophrenia. Also, it turns out that people who experience like anxiety attacks, panic attacks, psychosis after being exposed to cannabis, a single nucleotide polymorphism in COMT, catechol O-methyltransferase, is predictive of that, is predictive of if somebody might have like a negative psychiatric reaction to cannabis, which minority of the population, to be entirely clear, but it is one example that it's one of a few that have been found where people have aversive reactions to cannabis. Keep me one of those heady toppers. Yeah, sure. Yeah, you got it, man. And a focal banger. All right, that's what I got for you today. Once again, if you dig this kind of discussion, maybe you'll consider subscribing and liking and all that kind of stuff. Otherwise, I will see you next week. See you, everybody.